stem part, you have to create a cut. Due to that cut, now that area they secrete the traumatic acid. Traumatic acid, nothing but these are the one kind of the auxin. You know very well, auxin responsible for the root induction. And whenever that cut area, from that cut area, formation of the root. That root basically they are adventitious root because they arise from the stem area. And such kind of this technique simply it is called as the layering method. Now I'm talking about the another type of the layering method, that is air layering. That yesterday what we discussed, that is a soil layering, because we have to put that same part inside the soil. But now we are talking about in uh, inside the air or in exposure without soil, how we can create a new plantlet. So air layering, also called as the gutti. In case of the gutti, just check the chat box. I just shared one image. Normally what happen, for the air layering or for the gutti purpose, you have to select a mature and the hard stem. First of all, right? Should not, it does not requirement of the any soft and the flexible stem, because whenever we are talking about the air layering, also called as the gutti, that time soft and the flexible branch or the soft and the flexible stem part, it is not required. Sometimes what happen, it is required the hard stem, or simply you can say that is woody stem is required. First of all, second one, no requirement of, no requirement of soft and flexible stem. Okay, so just the technique, simple technique. Now in this point, suppose these are the part of the stem. Okay, part of the stem. From that stem area, these are the one branch. It will be arise. Now purposefully, you have to remove some part like this. You have to remove the bark region. So first step, remove bark. And you know very well, bark basically these are the outermost layer which is a dead part. Simply we are talking, हम आगे देखेंगे इस anatomy chapter में. So basically these are the bark. So you have to first step, that is remove the bark. Once remove get bark, bark it is get removed, ultimately whatever the vascular bundle is there, or whatever the vascular cambium is there, so that vascular cambium it will be get exposed, right? वस्कुलर कैम्बियम, ये भी हम आगे देखेंगे इन एनाटॉमी चैप्टर, व्हाट इज मीनिंग ऑफ द वस्कुलर कैम्बियम. बहुत सारे कैम्बियम्स होते हैं बेसिकली, लाइक कॉर कैम्बियम और द इंट्राफेसिकुलर कैम्बियम, इंटरफेसिकुलर कैम्बियम, रिंग कैम्बियम, वस्कुलर कैम्बियम. सो जस्ट इमेजिन वन थिंग, cambium means what? These are the something meristem who can form xylem, who can form phloem, who can form a different types of the cells. So we are talking about that is cambium part. It's like a meristem. Just suppose. So first step, that is you have to remove the bark region. Due to removal of bark, now whatever the vascular cambium that it will be get exposed. And now what happen? Whatever the exposed area, they should be covered with the help of grafting clay. Grafting clay. Okay, so it should be covered with the help of the grafting clay. So now what happens, simply we are using the clay. At this area we have to use the clay. Entire area they should be covered with the clay like this, entire area. So that area nothing but these are called as the grafting clay. And after the grafting clay, you have to cover with the some plastic paper, or you can use any wrapping paper, everything you can use anything. So normally one thing, these are the one kind of the layering technique where we require the woody stem, because we have to remove the bark. Once you remove the bark, vascular cambium they get exposed, and now that area we have to cover with the grafting clay. And once we form the, once we apply the grafting clay, now you should cover with the some plastic paper or something else, right? So we are talking about what is the exactly composition for the grafting clay, because inside the grafting clay something ingredients are present, and that's why they can induce root, they can induce root formation, and they can give rise to new plantlet. So that grafting clay, it has some composition. So one by one we are talking about the part of the composition. Inside the grafting clay, normally there is a presence of one part of moss. Can you tell me why we are using the moss? Anyone? What is the basic function of the moss? हमने देखा था. In case of the sphagnum, we already discussed this point. Why moss? One part of the moss. Attachment. Yes, it is to have. It's having good water holding capacity. Good. So they can hold the water. So it is used for the water holding process. Another one, again, one part of cow dung. And you know very well, cow dung basically these are the one natural fertilizer. So as a fertilizer, we can use cow dung. Again, you have to use two part of clay, okay, any clay, but that clay it should be having good binding capacity. So clay we are using as a binding agent because you know very well moss and cow dung ये तो चिपकने तो वाला नहीं है perfectly. So we have to create, we have to take the clay part. So when you mix it properly, so now it can create one, it's not it should be like a some, what we say, clay, it should be like a clay, and they can hold, they can exactly adhere that particular area. So two parts of the clay, and as well as you can add some amount of the water also. And most important thing, you have to add some root setting hormone. And root setting hormone nothing but you can use auxin, and in case of the auxin, two example, IBA, and sometime you can use NAA. एक second, I'll open the chat box. Okay. So one part moss, one part cow dung, two part clay. Again, some amount of the water also there. And most important thing we are talking about, there is a requirement of the auxin. And you know very well, we are purposefully creating a cut. We have to provide a nutrition because last technique में क्या हो रहा था? Plant naturally they can secrete the traumatic acid because traumatic acid these are the one type of the auxin. But now in this condition we have to use artificial auxin, means artificially synthesized auxin, which is not produced by the plant, which is synthesized in lab, which we prepared, right? So as an auxin source, you can select IBA, and the long form for the IBA, that is indole butyric acid. IBA, or another one you can select NAA, naphthalene acetic acid. Okay, so all these are the condition. And now what happened due to that auxin? Obviously, we are providing moss who having the good water holding capacity, cow dung, which is a natural fertilizer. So again, clay also provided as a binding agent, water also provided, and as well as the auxin also provided. So due to all those favorable conditions, now in that particular area, root induction it can initiate it. Root induction, and you know very well, once root get formed, ultimately there is a formation of the new plantlet from the entire region. So such kind of the technique, these are called as the gutti, also called as the air layering. कल वाला जो हमने देखा था ना, that is a natural method, that is the soil layering. Now we are talking about the air layering because whatever the plant is present, whatever the technique is performed, that they are exposed to air without soil. Okay, and simply such technique it is called as the gutti technique. And actually these are the very ancient technique, बहुत पहले से चला आ रहा है. People know this technique. So create a grafting clay, and these are the something composition, and you have to create, because what is the purpose? Root induction. And you know very well, if root arise from the stem part or any branches or any leaf part, such type of the root, they are called as the adventitious root because radical is not involved. Done? Got it? Any doubt from the gutti? Okay, now we are talking about the grafting. Second part, after the layering, very simple, straightforward, we are talking about the grafting. And during the grafting, one question you have to remember, what is meaning of scion, what is meaning of this stuff, and number third, where this grafting technique can be performed. So second technique we are talking about, that is grafting technique. For the grafting technique, most important thing, cambium is required. Cambium is required. It's must. And that's why you know very well cambium, they are present in case of the gymnosperm and as well as they present in case of the dicot. So dicot and the gymnosperm you can use as for the grafting technique because inside the gymno and the dicot plant cambium present. But please remember in monocot basically cambium is absent, right? So that's why for the grafting technique you never use monocot. Got it? So gymnosperm and the dicot you can use as an experimental material for the grafting. So what is meaning of the grafting? इसके बारे में बात करें. Suppose these are the two plants. Hmm? We are talking about two plants. Suppose these are the plant A and now we are talking about the plant B. It's something like you have to create a superior variety like that. So suppose we are talking about these are the part A, means plant A. Inside the plant A, it has some uh, good quality of the shoot. So we are talking about it has shoot superior. Whatever the shoot is present, it is having the shoot in plant A. But now what happened? Another problem, it has root, but root it is a quite weak. So you know very well for the healthy plant, root should be strong because via root mineral water can be absorbed. Another one, shoots should be strong because with the help of the shoot, photosynthesis, that is food synthesis can be performed. So in any particular plant, दोनों system strong होने चाहिए, shoot system as well as the root system. But now what happened? There is one drawback. Plant A it has superior shoot, but it has the weak root. Exactly reverse scenario with respect to plant B. In case of the plant B, now shoot basically they are weak and root part now they are strong, that is superior. So with the help of the grafting, you can perform a third variety, right? Combination karo. And that time what happen? You can perform a one new variety and with the help of that variety, clearly you can say that is both the qualities, good qualities, that is shoot as well as the root. Both they are obtained superior quality. Both they can obtain the superior quality. So that time you have to use which technique? Grafting technique. So I'm talking about the one, two words. What is meaning of the scion and what is meaning of the stalk? Okay. Now, in case of the plant A, definitely we are talking about which part is superior. Which part is superior? Shoot part is superior. So you have to select which part? You have to select shoot part. So shoot select and shoot simply whatever the part which is used for the grafting process. That part is called as the scion. Shoot part simply it is called as the which part, that is scion. Okay. In plant B, which is the superior one? Root superior है, obviously. So you have to select the root. Just check your mic. इसका mic on है. You can select the root part. And now what happened? Whatever the root part is there, whatever the particle we selected, that part simply it is called as the stalk. That called as the stalk. So now what happened? You can create a V cut, you can create a oblique cut, now you can create a multiple cut, बहुत सारे cutting के pattern है. But now what happened? Plant A, you have to select one stem like this. And plant B, you have to select the another part. You can create a V cut and with the help of the V cut, ultimately you can join with the grafting clay or the grafting paper, you can wrap it and ultimately what happened? Obviously we are create a particular cut, cambium they get exposed, cambium they undergo the self division process and they can initiate, they can join entire xylem phloem, they can join with each other. So this green color, simply we are talking about these are the part of the shoot, that shoot part simply it is called as the shoot of plant A, which has the superior quality. Lower part basically we are talking about the stalk nothing but these are the root part and root part superior part coming from the plant B. So now what happened? You have to create a V cut on both the scion also we have to create a cut, as well as the stalk also we have to create a cut. You have to adjust, because see जब कभी भी आपको क्या करना है, the process like grafting process, so that time scion and this stalk both they are having same diameter. Obviously अगर एक छोटा होगा एक बड़ा होगा, that is very difficult to fix them. So whenever you have to perform any process like grafting process, so that time you have to take one precaution, that is scion and this stalk they are having same diameter. So that time you can successfully create one kind of the new creation, and whatever the area which is get exposed, that area you should be covered with grafting clay. And just we discuss what is the composition of the grafting clay. Simple technique. Such technique simply it is called as the which technique? Grafting technique. You have to select the scion from the shoot, stalk from the root, create a cut, attach them, covered with the grafting clay, and after 2 months or 3 months from those areas, means xylem, phloem, there is a formation of the new cambium, and now you can obtain one superior quality of the plant, that is called as the which one? Grafting plant. मोस्टली सच टेक्निक इज वेरी मच कॉमन इन केस ऑफ दी रोज. आपने देखा होगा रोज में बहुत बार मल्टी कलर के भी फ्लावर्स लगते हैं. समटाइम दे आर हैविंग समथिंग कॉम्बिनेशन, मींस रेड कलर भी है, मींस व्हाइट कलर एंड द रेड कलर यू कैन मिक्स एंड यू कैन ऑब्टेन द पिंक कलर. सो सच काइंड ऑफ दिस कंडीशन आल्सो प्रेजेंट, right? So it's very much common. Okay, so when we are talking about such grafting technique, for the grafting technique first of all you require the cambium, and inside the cambium gymnosperm and the dicot you can use. In case of the monocot, no cambium present, so for grafting technique you can never use monocot. Got it? Simple, straightforward. Any doubt from the grafting? इसके एक्चुअली बहुत सारे टाइप्स भी होते हैं, बट no need actually for one question, no need honestly. Okay, yeah, any doubt boys? Edric? Okay, crystal. Yeah, next point, very simple, we are talking about the cutting, last one. Number third, we are talking about the cutting. You know very well, cutting nothing but you have to create a cut, create a cut on plant part. Like you can select stem region, you can select root region, or you can select leaf region, any part region. Okay. And now when you create that cut, lower part it should be dipped into oxime solution. So for example, whatever we create a cut, that area it should be keep under the oxime solution, and you know very well, once due to oxime, now that root get initiated, and there is a formation of the new plantlet. So इतना ज़्यादा important नहीं, यहाँ से question नहीं बनेगा. Whatever the question coming, one question from the gutti and the another question from the grafting. Cutting means what? Simply जैसे हम hibiscus plant also we are create one slanting position cut, and sometime what happen, you have to use, you directly you can put inside the soil, so they can also form a root. That is a natural method. But in case of the artificial method, you have to use some concentration of the oxime. You can dip for the 5 minutes, you can dip for the 30 minutes or 1 hour or 1 day. It depends upon the plant to plant. So simply create a cut, dip into oxime solution, and they can responsible for root initiation. Done? Yes. So these are the all information about the artificial reproduction, which is a part of vegetative reproduction. So we natural भी देख रहे, and we cover artificial also. चलो, done? Done? Now we are talking about the another important question, which is a part of the sexual reproduction. I'm not taking too much detail because it's Zoom में भी repeat होगा. Again ये repeat होगा in sexual reproduction of the flowering plant. So we are focusing two important thing. Number one, that is Strobilanthus kunthiana, and the another one that is bamboo, because वहां से questions बनते हैं. So next part we are talking about sexual reproduction. Last point of this chapter. You know very well, sexual reproduction basically it's a biparental, gamete formation भी होगा, gamete fusion भी होगा, and after the gamete formation also present, it's a biparental, two parents they get involved, and after the gamete formation, now there is a gamete fusion also present, right? That is called as a fertilization. And after the fertilization, you know very well, male gamete they fused with the female, and finally there is a formation of zygote. And zygote they are always and always diploid in nature, always and always diploid in nature. And you know very well, zygote again, these are the vital link between parent and new offspring. These are the vital link, important link between parents and offspring, new offspring, parents and new offspring. That's it. Another one, it's a very slow method, very complex method, and number of the progeny also less. वहां से question नहीं बनेगा. So one question, that is it is always diploid. Another one, these are the vital link between parents and the new offspring. Now we are talking about during the sexual reproduction, whatever the phases are there, means we are talking about the life cycle phases. These are the controlled by the hormone. Even in case of the plant also they are controlled by the hormone. In case of the animal also it is controlled by the hormone. So next point, we are talking about different phases, life phases actually, different phases in life cycle. And please remember this point, whatever we are talking about, all these phases, these are the controlled by hormone, even in case of the plant as well as in case of animal also. Three important phases are there. Number one, there is pre-reproductive phase, that is also called as the juvenile stage, or pre-reproductive phase. During the pre-reproductive phase, plant maximum they are showing vegetative reproduction. They are showing vegetative reproduction because sexual part, male gamete, female gamete, anther, or whatever we are talking about, that is gynoecium, they are not developed. So whatever the developmental parameter is there, they are all showing the vegetative reproduction. Number second, now that plant it is become reproductive, and you know very well, whenever we are talking about the reproductive, it means that the sex organ they are showing maturity. So maturity of sex organ, so they are capable for the fertilization process, they can form a new offspring. And finally we are talking about last stage, that is senescence, programmed cell death, or simply we are talking about post reproductive phase. Post reproductive phase, right? That is it's towards to death. So when we are talking about the post reproductive phase, ultimately whatever the metabolic activities are there, जितने कुछ भी हो रहा है, replication, transcription, translation, or everything, metabolic activity, catabolism, anabolism, all those process ultimately they get decreased, and finally death will be occur, that is programmed cell death, natural death actually, not accidental death. So during the different phases of the life cycle, first one that is juvenile stage, another one that is reproductive stage, and the last we are talking about post reproductive phase. On the basis of that different phases of the life cycle, we are taking two important concepts. Monocarpic means what, and polycarpic means what, means flowering and fruiting once हो रहा है, and or it is happening repeatedly, means after 10 years, after 12 years, or every year flowering and the fruiting. So we are talking about those points on the basis of life cycle. Should I continue? Should I continue? Ramchandra, Pratyusha, Nihal, Komal, Abhilasha, Gargi. Okay. So ये point important है for your neat point of view. Now, another one we are talking about. Please divide your page into two parts. Now we are talking about monocarpic, and this compare with polycarpic. Monocarpic and polycarpic. Simple meaning. Whenever we are talking about the monocarpic means what? Flowering and fruiting once. Flowering as well as fruiting. पूरे life cycle में only once. That's why it is called as the monocarpic. But when you compare this point with polycarpic, it means that regular after the regular interval, again and again flowering and fruiting. So flowering and fruiting basically they form after regular interval. Flowering, fruiting, they occur after regular interval. ठीक है, गैप होगा एक्चुअली इसमें, बट इट हैज सम, मतलब 1 टाइम 2 टाइम 3 टाइम और अप टू डेथ अगेन फ्लावरिंग एंड फ्रूटिंग इज कंटिन्यूअस. वो फर्स्ट पॉइंट. Number second point, when we are talking about monocarpic condition, so this very important, you have to remember example. In case of the monocarpic condition, first one, all annual plants, these are the example, and you can write down the example. Rice as well as the wheat, they are come under the annual plant category, as well as all biennial plant. Biennial plant. In case of the biennial plant, again you can remember the example, that is radish, then carrot and henbin. Henbin. So annual plant means what? During the vegetative growth means all the three phases like juvenile phase, reproductive phase as well as the post-reproductive phase, all these three phases they occur within 1 year. So such type of this plant, these are called as the annual plant. Biennial plant means what? Visit all these three phases like reproductive, then pre-reproductive and the post-reproductive, all those life cycle they can complete within 2 years. So such type of the plant, these are called as the biennial plant. And in this category, in case of the monocarpic, you have to remember few perennial plants also. Few perennial plants also. And one important question for the NEET, perennial plants means what? The plant, those are showing slow but the prolonged growth. And in this point of view, we are taking example bamboo species. Bamboo species. Very important step, very important sentence for the bamboo species. Inside the bamboo species, flowering occur after 50 to 100 years. So can you imagine perennial means what? The plant are showing the slow growth. So imagine करो कितना ज़्यादा इसका slow growth है. Reproductive phase is very larger actually, and once they flower, एक बार ही flower होगा, and then उसके बाद they undergo the apoptosis, they undergo the senescence process. So in case of the bamboo, we are talking about flowering once hoga, obviously, because it is a part of the monocarpic. But after flowering it will be occur between 50 to 100 years. So can you imagine how much it's a long period? And that's why bamboo, they are example of the perennial plant, because they are showing continuous but slow growth, slow prolonged growth, such type of the plant called as the perennial plant. So bamboo, it is an example because they are showing flowering after 50 and 100 years. Okay. So please remember this point. Whenever we are talking about the monocarpic means what? Whatever we are talking about the three life cycle, that is juvenile phase, reproductive phase, and the senescence phase, that is post-reproductive phase. They are occur within 1 year, that is annual, 2 years, biennial, or sometimes they are showing prolonged like few like perennials, which is example of the bamboo. But एक बारी flower होगा. That's it. Okay, only once. That's what is called as the monocarpic. Got it? Yeah. And now we are talking about next example for the polycarpic condition. So now what happened during the polycarpic condition? First of all, there is a presence of the juvenile stage, that is pre-reproductive. First of all, there is a pre-reproductive. Again, they can enter during the reproductive phase. After the reproductive phase, now there is a gap, small gap hoga. So for example, during the reproductive phase, now these are the first time formation of the fruit and the flower. So for example, these are the first fruit. Uske baad, again it will be undergo the process of the reproduction. Yes, Pratyusha. But during this, now they can form a second fruit. But now what happened? Between that first fruit formation and the second fruit formation, यहाँ पे थोड़ा सा present होगा gap. There is a gap is present, right? So why these are the gap is present? Because it's one kind of the recovery phase. Because just plant they are form of fruit, and you know very well, fruit formation means obviously they are providing nutrition, right? So there is a small gap is required between two reproduction. There's a small gap is required, so that's why this gap means repeatedly reproduction over it doesn't, it's depend upon plant to plant, right? But now in this condition what happened? Pre-reproductive, again reproduction, small gap, again reproduction, small gap, and finally death. Depend upon their lifespan of that particular plant. Finally death, right? Such kind of this condition. Regular interval में क्या हो रहा है? There is a formation of the fruit, formation of the flowering, obvious है. And such kind of this condition, it's very much common. We are taking examples right now. Just first of all complete this writing part. Once you done, please tell me, okay? Achal, Atul, Chirag. Okay. So now we are taking the another important question, neat point of view, that is example. जो common example you know very well, that is Nilkhuranji. You know very well there is a Nilgiri Hills. Nilgiri Hills, why we are talking about the Nilgiri? Because they can cover, Nilgiri Hills, they can cover Kerala region, Karnataka region, as well as the Tamil Nadu region. And at the particular year, after the 12 years, all those Nilgiri Hills, it is converted into blue stretches. They entire, they look like a blue color. Agar image just take the image and share it inside the chat box quickly. So in this condition, what happened? We are talking about very neat favorite actually example, that is Strobilanthus kunthiana. Kunthiana. And common name these are also called as Nilkhurinji. Okay, blue color flowers, very attractive flowers. Blue color flowers. Normally, what happen? These are the Kunthiana, Strobilanthus kunthiana, also called as a Nilkhurinji. They are showing flowering once after 12 years. They are showing flowering after every 12 years. So bamboo 50 to 100 years and Kunthiana after 12 years, right? And now what happened? एक साथ ही फ्लावर होते हैं, so that's why जितना भी एरिया है नीलगिरी हिल्स का, so that entire area is converted into blue color. So mass flowering, actually mass flowering, एक दो plant नहीं होगा वहाँ पे, entire means million trillions plants are there and all those are showing at one time flowering, so that's why it look very beautiful. And example, that is who covered the Nilgiri Hills area. Nilgiri Hills means we are talking about Kerala, Karnataka, Tamil Nadu area, where all the hills region they converted into blue color due to flower Kunthiana, Strobilanthus kunthiana. Okay, that's it. So it is example for first example for polycarpic condition, and the another example we are talking about perennial also. So in case of the perennial plant, you know very well, mango. Every year we eat mango because it's a seasonal, and as well as the apple also, apples and the mango. So यहाँ से question नहीं बनेगा actually apple and the mango, but you have to remember Strobilanthus kunthiana. Okay, yeah, done. Yeah, thank you. Yeah, it's exactly Pratyusha. It's very beautiful. अगर chance मिला तो जाना देखने. हाँ, मैं बता देती हूँ. हाँ, one thing according to NCERT, अकॉर्डिंग टू एनसीईआरटी जो हमारा बुक है, लास्ट टाइम फ्लावरिंग हुआ था 2010 2006 में, सितंबर अक्टूबर समथिंग, इट्स अकॉर्डिंग टू बुक. बट इफ आई एम टॉकिंग अबाउट द एक्चुअल कंडीशन, करंट कंडीशन, इन 2018 में फ्लावरिंग हुआ था. दीज आर दी अक्टूबर करंट कंडीशन, बट जस्ट यू हैव टू फॉलो द बुक बिकॉज हमें बुक के हिसाब से ही जाना है. So according to book 2006 they are recorded for the Kunthiana flowering season, but actually these are the 2018. तो imagine करो after 12 years मतलब कब आएगा ये, in 2030. Yeah, चलो. So any doubt from the monocarpic and the polycarpic? Any doubt? Okay. So whatever the part related to gametogenesis, because whenever we are talking about the fertilization process, again these are the three phases actually. Number one, that is pre-fertilization event, that is gametogenesis, formation of the gamete, then fertilization event, and then after fertilization, that is post-fertilization event, means zygote form hoga, embryo form hoga. So that is not important. Hmm? Next one we are talking about, last point, two important condition. Sometimes question will be asked. So we are talking about what condition, means monoecious condition and the dioecious condition example. So we are talking about first of all the example for the dioecious plant. You know very well dioecious plant means what? Whatever the sexual organs are present, they present on the two different plants, right? So dioecious. So in case of the dioecious, we already discussed one example, Merchantia. Remember, all bryophytes they are monoecious in nature except Merchantia. Because Merchantia, these are the belonging to Bryophyta and these are the example for the dioecious plant. Another one, mulberry. Mulberry also they are example for the dioecious. Now papaya also they are example for the dioecious. Date palm also example for the dioecious plant. And cannabis also they are example for the dioecious plant. Another one condition we are talking about, monoecious plant. Monoecious means what? Both sexual part they are present on the same plant. तो हमने पहले ही लिस्ट देखा हुआ था, क्विकली वी आर रिवाइजिंग दिस पार्ट. अंडर द मोनोशियस हमने देखा था कोकोनट, कुकुरबिट्स, बट आई एम टेकिंग अ वेरी यूनिक example which is belonging to lower plant, that is Kara. I hope you remember Kara basically it is example for the green alga. ये exception case में भी आ रहा था because Kara, inside the Kara, whatever the sex organs are there, all the sex organs they are multicellular plus jacketed. In case of the algae, in all algae, sex organs they are very simple without jacket and they are unicellular. But exception case में हमने देखा था because Kara is the example for the exception case. And what kind of the exception condition is present? In this condition, whatever the sex organs are present, that sex organ, they are jacketed. Jacketed means what? Protective layer is present around the sex organ. So they are jacketed plus they are multicellular. Normally uni hote, but in this condition they are multicellular. Right? So I'll just draw a diagram, very simple diagram, because I just want to tell you exactly position, because it is an example for the monoecious plant. Means what? Male and the female, they present on the same plant, but same, but kahan pe? Right? So we are taking diagrammatic representation for the Kara. हम ज़्यादा डिटेल में नहीं जा रहे, I'm taking very simple diagram. ठीक है? Now we are selecting this part near to the node. So we are selecting this part. Suppose now in this area we are taking some highlight. Towards the upper side, please remember, toward the upper side, now in this condition, oogonium is present. And oogonium is what? We are talking about female reproductive part. Oogonium. Where this present? Upper side. And exactly below, now there is a presence of the male. It's lagging? Okay, I just wait. Yeah. Now it's visible. I just draw some small diagram actually, because I just want to tell you where the exactly male and the female is present. Upper and the lower, आपको उतना ही याद रखना है. That's it. Now, okay. Just wait. I'll share screen again. Huh? I'll stop it and again I'll share it. I think it's visible, right? Okay. Thank you so much. So just quickly, we are talking about one example for the monoecious plant, that is Cara, which is example of the green algae. Exception case है because in case of the Cara, basically sex organ, they are jacketed plus multicellular. So we are selecting one node area, that is leaf area. And whenever we are talking about toward the upper side, sex organs are present. If I'm talking about which sex organ, so female, they produce toward the upper side. And exactly near the female, now there is a presence, lower side, presence of the male. And male, nothing but we are talking about antheridium. Right? There is a presence of the antheridium. That's it. So you have to remember in case of the Kara, if is diagrammatic structure toward the upper side, female structure is present, female reproductive part, and toward the lower side, male reproductive part is present. That's it. Done? Done, guys? Okay, Gargi. Chalo. Last point. Fertilization internal and the external. So you know very well there is a two types of the fertilization. Fertilization may be internal or external. So during the external fertilization, now there is a risk. Risk is what? Because there is a exactly chance phenomena. So whatever the male and the female gamete they produce, they have to produce in a large amount because simply male and the female gamete they release inside the water और वो बाकी सब कुछ chance phenomena पे water जहाँ पे move करेगा along with water movement. Now that male gamete and female gamete they randomly get fused. So that's why during the external fertilization, bulk amount in high amount male and the female gamete we should release, right? And please remember external fertilization it is mostly present in case of the algae, mostly, but in case of the fungi also, not all, few fungi, such type of the external fertilization is present. And you know very well internal fertilization means what? Obviously during this condition male whatever the male gametes they are present or pollen grain they should present in a high amount, but female they are present in a limited number, egg should be limited number. So now what happens, it can be compensate or during the internal fertilization the chance of fertilization to be increased because exactly position पे male gamete fused होता है. Male gamete they can easily transfer toward the female and mostly inside the female body the sexual reproduction process can be occur. So external, so fertilization is related to algae, but internal fertilization which is a more what we say healthy way for the fertilization where the chance of fertilization is get increased, and it is present in all bryo, it is present in all pterido, it is present in all gymno, and as well as it is also present in all angio. That's it. Hmm? Chalo. So with this, now there is the end of this chapter. That's it. Any doubt from this chapter? Any doubt? Smartly study करना है. Once you start plant kingdom biological classification, then you can start with the plant kingdom, then continue with the morphology, and immediately you can start this chapter reproduction. बहुत कम टाइम में बहुत कुछ रिवाइज होता है, but you have to perform a smart work. Okay, and one question coming from this chapter, only one. So it's your strategy how you are using this. Next one, very important chapter, major chapter, and the common chapter for the botany as well as the zoo because cell present everywhere. Cell it is a part of the bacterial body, cell it is a part of the animal, cell is a part of the plant. So whenever we are talking about the living entities, that time we can say cell. So next chapter, cell, the unit of life. Okay, and for the upcoming test syllabus I already declared entire morphology chapter. Please don't consider reproduction because जो reproduction का अभी जो हमने part complete किया, I will merge that chapter in sexual reproduction in flowering plant. So for the upcoming test only one chapter, morphology in flowering plant. Okay, yeah. So we are talking about say the unit of life. Again Another neat 2019 question. बहुत कुछ, if you read the module, you will get so many number of the scientists' name, but you have to remember only 5 names of the scientists. That's it. So first of all, we are talking about historical account. It's very easy chapter, okay? And this chapter only 1 or 2 times it's required revision. बहुत कम time में इसका revision हो जाता है, because again from the lower classes you know all these cells, but just strategy. So first of all, we are talking about some historical account. The contribution, first scientist, Robert Hooke. This chapter 2 to 3 questions. Pratyusha, cell chapter 2 to 3 questions. If I am talking about the entirely cell unit of life and the cell cycle, cell division, totally 5 to 6 questions. So that's why once I started the cell, I'll end cell cycle, cell division also, because दोनों के दोनों overlapping है, means connected with each other. So total 6 questions from both chapter. So देखो 6 into 4, it's major chapter. Okay. So first of all, we are talking about the first scientist, that is scientist Robert Hooke. Scientist Robert Hooke first time identified dead cell, not living cell, okay? First time identify the dead cell. So you know very well, he just collect the glasses and he placed in a series, and after the series he just observed the cork cell, and you know very well, he was selected a thin slice of cork, and under the microscope he just simply observed something, a honeycomb like structure, and he considered that yes, it is a cell according to scientist. Okay, so Robert Hooke, direct question, first time identify the cell, but cell was dead. One question. Another point, scientist Leeuwenhoek. Scientist Leeuwenhoek, now he identified a living cell. Identified a living cell. So first time he observed the bacteria, then second time he observed the protozoa, RBC also he observed, spermatozoa also observed. But living cell discovery, contribution or the credit gives to scientist Leeuwenhoek. Third scientist Robert Brown. Robert Brown, गड़बड़ मत करना, Robert Hooke different है, Robert Brown. First time Robert Brown observed and they discovered nucleus from the orchid root cell. So first time. So he identified and they discovered nucleus. So obviously, whatever the scientists Robert Hooke and the Leeuwenhoek, they just doing the morphological study, right? It's moving or it's not moving. But first time, scientist Robert Brown, he observed nucleus, which is an internal part of the cell. So nucleus it is get discovered, and such nucleus discovered from orchid root cell. From orchid root cells. Okay, now next one, two scientists. One scientist Claydon and the Swan. Both their friends. Claydon and the another one scientist, that is Swan. Claydon basically he was a botanist and Swan basically a zoologist. So both they give contribution. So first of all, we are talking about this Claydon. Claydon explained that all plant cells they are made up by more than one cell. That is also true, because normally you know very well, unicellular cell also present, like in case of the algae, and multicellular cells organisms also present, like bryo, pterido, gymno, angio. So what Claydon explained, he simply explained whatever we are talking about, all plants they made up by one or more than one cell. One or more than one cell. That is also true, because हमने देखा था. One cell means we are talking about the algae, and the more than one means bryo, pterido, gymno, angio, everything. So one cell you can remember as a algae, and more than one cell, that is bryophyta, pteridophyta, gymnosperm, as well as the angiosperm. That is also true. Then after few years, scientist Swan also explains such condition. So he just said, explained one condition, that is in several animals. In several animals also, they are made up by the cells. They made up of cells. He observed many cells. He are not talking about the all. Please remember, उसने all plant बोला था. In case of this Claydon, he explained all plant. But scientist Swan explained several animals. In several animals, he simply said that yes, body is made up of the cells. And again, he explained one another important scenario, that is in case of the animal, cell wall is absent. That is totally true. Cell wall is absent. So these are the again important scenario, important explanation given by the scientist Swan. So four scientists. Number one, Robert Hooke, Leeuwenhoek, Robert Brown, Sladen, and the Swan. Okay. Now we are talking about the another important point with respect to cell theory. What is meaning of the cell theory? Should I continue? Make your notes, okay, because if you read from the module or the NCERT book आपको कुछ समझ में नहीं आएगा, because NCERT से cell chapter के question design नहीं होते, you know very well. If you practice more question, then you can understand. If you solve the previous year question, you will get clear cut idea. From NCERT, you never solve question from the cell chapter, so please make your notes properly. Now, so next part, after these four scientists, we are talking about cell theory. And this cell theory again given by two scientists, two friend, best friend, that is Claydon and the Swan. Okay, so we are talking about which important points which is a part of the cell theory. Number one, according to two scientists, cell is a structural unit of life. True. ये तो definition ही है cell की. Cell is a structural unit of life because obviously each and every cell, all living organisms, they are composed by cell and their product. For example, cell hoga, inside the cell enzyme present, protein present, cell organelles are present. For example, chloroplast, so that chloroplast is having a specific type of the structure. Obviously, the number one point also true. According to cell theory, cell is a structural unit of life. Another point, cell is a functional unit of life. That is also true. Cell is a functional unit of life. That is also true because you know very well all activities which is occur inside the organism, which is occur inside the cell, they are very specific. Like inside the cell, chloroplast is present. So that chloroplast, they are performing a specific type of the function like photosynthesis. Inside the cell, mitochondria presented, powerhouse of the cell. They perform the function that is ATP synthesis. So each and every cell, they have a specific type of the function. So yes, cell is a structural unit of life as well as they are functional unit of life. Another point, if I'm talking about the any cell, it is consist of protoplasm. That is also true. Cell, it is a consist of protoplasm, and inside the protoplasm, two important components are there: cytoplasm plus nucleoplasm. Cytoplasm plus nucleoplasm. That is also true. Suppose we are taking one example. These are the egg cell. These are the egg cells. Suppose inside the egg, now there is a presence of the part of the nucleus. Outermost boundary, these are the plasma membrane. For the plasma membrane, for further reference, I will use PM. Please remember, okay? For the cell wall, I use CW. For same, for the plasma membrane, I will use short form, that is PM. So these are the PM, that is plasma membrane. Now, ये जो part है, that is nucleus. Inside the nucleus, whatever the material is present, whatever the fluid material is present, that fluid material, it is simply called as the nucleoplasm. So you know very well, inside the nucleus, genetic material present hoga, histone protein present hoga, non-histone proteins also present, RNA present hoga, nucleus also present, so chromatin, everything. So whatever the structure is present inside the nucleus, all those fluid part, that is called as nucleoplasm. And outside the nucleus, रोज इसी टाइम पे आता है. Okay. So whatever the material which is present, you have 2 seconds please. गिव मी योर एड्रेस, मैं उसको तुम्हारे घर भेजती हूँ रामचंद्र. ठीक है, आ जाएगा बराबर पम पम करते. Okay, chalo, finally. So whatever the fluid material is present, which is present outside the nucleus, that material is called as the cytoplasm. So obviously we are talking about nucleoplasm. Another one, that is cytoplasm. Again, inside the cytoplasm, cell organelles are present. Again, water is present, other fluid material, enzymes, protein, RNA, बहुत कुछ present होगा. So collectively they are called as the protoplasm. So yes, whenever we are talking about any cell, that any cell, it is consist of the cytoplasm as well as the nucleoplasm. And the last point with respect to cell theory, that is all cells, they are basically they having same in structure as well as their same in activities. तो ये हम point पहले cover कर चुके, that is structural and the functional unit of the life. So just remember only 3 points. Now, such cell theory, it's true, but it has some drawback. So we are talking about what is the drawback of this theory. So one drawback, basically, scientist Clayton and this Swan explain everything very nicely. That is also true, but he failed to explain how that new cell they get arise. So there is a no information, no info. How a new cells it will be get arise, right? And इसका solution, इसका answer दिया the scientist virtue. We are talking about scientist given by Rudolf Virtue. NEET 2019 question. Scientist Virtue, basically he explained one point, that is new cells arise from pre-existing cell. He explained, he gave the reason, what is the drawback, he recovered that, and he explained that is new cells simply they are arise from pre-existing cell. Obviously हमें पता ही है something a mother cell is present, something whatever we are talking about parental cells are present, and from parental cell now there is a formation of the new cells. But scientists, according to cell theory, two scientists they failed, but scientist Virtue explained that point, that is new cells arise from pre-existing cells, and it is explained by two words, that is omnis cellula e cellula. प्रत्युषा कौन सी लैंग्वेज है ये? So it is explained by one sentence, that is omnis cellula e cellula. And what is the meaning of that? Yes, it is a Latin. So what is the meaning of that omnis cellula e cellula? That is new cells, they arise from pre-existing cells. That's it. Given by scientist Virtue. Done? So from the historical account, one question, please remember 3 questions bante. Last, if I'm talking about last year, 2 questions coming from the eukaryotic cell and the 1 question coming from the prokaryotic cell. And in this chapter, what we did means prokaryotic cell, it is again we repeated here because in kingdom monera bhi humne dekha tha prokaryotic cell. Here also all those points again repeated. So it's very easy. Chalo. So all these are the historical account. You have to remember only 4 to 5 scientist name. That's it, and 1 question from scientist name. Done. Okay. Chalo. Next point we are talking about cell size. You know very well, bacteria basically belonging to kingdom monera. They are prokaryotic cell, and obviously on the basis of size, bacteria it has 3 to 5 mu m by their length. 3 to 5. But meet favorite bacteria, mycoplasm. Mycoplasm, also called as the PPLO. Mycoplasm, it has size only 0.3 mu m. And such mycoplasm, basically they are smallest bacteria. It is example for the smallest bacteria. I hope you remember हमने बहुत कुछ देखा था related to mycoplasm. Again, this we cover this part here also because mycoplasm is example of the smallest bacteria having only 0.3 mm in size. Yes, these are the jokers of the plant kingdom because mycoplasm they lack the cell wall. They can change their shape. Another one, in all bacteria, circular genetic material is present, but in case of the mycoplasm, genetic material it is linear. We'll discuss that part later commonly. So one question, mycoplasm. Another point we are talking about, normally we are taking one largest cell and you know very well, ostrich egg, it is example for the largest cell. So no need to remember size, but एक ही size आपको याद रखनी है mycoplasm, 0.3 mu m. बाकी का भूल जाओ. So ostrich cell, basically these are the largest one. Now when we are talking about nerve cell, that is neuron. So neuron cell, it is example by longest cell. So please get confused. Don't get confused. We are talking about the largest means what? Diameter wise. And nerve cell, these are the longest means we are talking about length wise. Done? Okay. Now, so next point, we are talking about metabolically active cell. And ये point जो होगा, this point is similar to cell cycle cell division also, so that during the cell cycle cell division, I will not explain this point. जो सुनना है यही पे सुनो. Metabolically active cell. So what is meaning of the metabolically active cell? Cells, they are showing more catabolism, more anabolism. Cell division it is showing rapid. So normally these are called as a metabolically active cell. But please remember, when we are using the word metabolically active, that time generally small size cell, they are more metabolically active as compared to large size as compared to mature one. So these are the exactly rule. So generally small size cell, they are metabolically active. So we are talking about the reason why such condition is present. First reason we are talking about, in case of the small cell or in case of the metabolically active cell, nucleus plus cytoplasmic ratio is high. Nucleus by cytoplasmic ratio is basically high. One thing. So we are talking about ratio high. It means that nucleus amount or nucleus ratio it is more as compared to cytoplasmic. Why such condition is happen? Because whenever we are talking about the cell unit of life, inside the cell unit of life, nucleus basically it is a brain of the cell. When we are talking about the nucleus basically it is a brain of cell. And normally what happen? Such nucleus they can control metabolic activity of the cell. So they can control metabolic activities of the cell. So ये भी point हम देखेंगे when we are talking about the anatomy chapter. In case of the meristem, normally meristem they are small in size and they are more active, because now what happened? Large small size cell, metabolically active cell, they having larger nucleus, dense cytoplasm, but nucleus size is quite large, because nucleus it is a brain of the cell. They can control all metabolic activity. So please remember, whenever we are talking about the nucleus plus cytoplasmic ratio is high, when we are considering cell, these are the small size, or cell basically they are metabolically active site, active. Okay. And we are talking about surface area by volume ratio. Could you tell me ratio क्या होगा, high or low? Any guess? Surface area by volume ratio is high or low? Quickly tell me. Surface area by volume ratio, what do you think? क्या होगा? Again, these are the high. Yes, these are the high, because what happen if surface area is more? If surface area is more, so ultimately what happen? Exchange of material more. Exchange of material, the rate of exchange of material, ultimately they can increase. So normally I'll tell you one thing. Suppose meristem. Meristem is what? Very young, metabolically active and very small cells. Normally what happen? Whenever we are using the word meristem, young hai, cell division rate is very high. But now what happened? The meristem cell or the small size cell, they have more surface area as compared to volume. Because now what happened? If surface area is more, because young cells hai, small size cell hai, it means that they are not capable for photosynthesis. So now whatever the food requirement, all those food requirement, it is fulfilled by the surrounding cell. तो सेंटर में एक मेरिस्टेम सेल प्रेजेंट है, स्मॉल साइज सेल, और उसके पूरे सराउंडिंग में मैच्योर सेल्स प्रेजेंट है. सो ऑल दोस मैच्योर सेल दे प्रोवाइड फूड टू स्मॉल साइज सेल. सो ऑब्वियसली absorption करना पड़ेगा, absorption of water, absorption of mineral, absorption of the food. So that's why for the absorption purpose, for the uptake, for the exchange of the material, small size cells, those are metabolically active, they having more surface area. Because when we are talking about the small size cell, no chloroplast present, no any plastid present, no any proper growth development is occurred. So that's why whenever we are talking about the such condition, number one, nucleus cytoplasmic ratio, it should be high, because if nucleus present in large amount, they can control metabolic activity. Another one, surface area volume ratio also high, because if surface area is more, so young cells, small cells, they are unable to perform a photosynthesis. So due to high surface area, they can easily uptake food, water, mineral from the neighboring cells, from the surrounding cells. Got it? Yes? Clear this point? ये पॉइंट एनाटॉमी में रिपीट होगा, ये पॉइंट सेल साइकिल सेल डिवीजन में भी रिपीट होगा. So please, if you have any doubt, you can ask me right now. Remember this thing, small size cell, they are metabolically active cell. Okay, chalo. Next part. Before starting prokaryotic and eukaryotic cell, now we are talking about difference between pro and eu, and we are taking only important point, because बहुत कुछ important stuff are there between the pro and eu, but no need. So we are taking only and only important difference between the prokaryotic and the eukaryotic cell. So please divide your page into two sections, and one by one we'll talk about major point. Prokaryotic cell, we compare this point with eukaryotic cell. First of all, number 1 point, cell wall. You know very well, prokaryotic cell, nothing but we are talking about, that is what we say, that is bacterial cell. Inside the bacterial cell, whatever the cell wall is present, that is non-cellulosic cell wall. Cellulose to present nahi hoga, definitely. What composition is present? We'll discuss that part later. So first of all, we are talking about there is a presence of the non-cellulosic cell wall. So for the cell wall, I'm using CW for the simplicity. So non-cellulosic cell wall is present. But we are taking one exception case, that is smallest bacteria, mycoplasm. So mycoplasm, these are the example for the bacteria where cell wall is absent. So for the absent, I'm using minus sign. अगर आपको word में लिखना है, you can. Okay. So question. All bacteria, they having the cell wall. Yes, cell wall is present, but that cell wall, these are the non-cellulosic cell. Non-cellulosic cell wall is present. But the mycoplasm, clearly cell wall is totally absent. Now, when you compare this point with the eukaryotic cell, we say in case of the eukaryotic cell, first of all, we are talking about plant cell. In case of the plant cell, whatever the layer is present, that is cell wall. That cell wall, it is made up by the cellulose, which is a major component. So cellulosic cell wall is present. In case of the protista, also, cell wall is present. So I'm writing just present. That is cell wall present, because in case of the protista, we always read those parts. In the protista, several type of the cell wall are present. Cell wall, it is made up of the cellulose plus pectin. Cell wall, they have silica also. Cell wall, it is rich with the protein also. So several conditions are present. So please for this refer kingdom monera, kingdom protista. So cell wall present in case of the protista. Another condition we are talking about in case of the fungi, yes, cell wall is present and that is made up by the chitin. So I'm just writing the word plus because you know very well, we already studied this part in kingdom fungi. Cell wall, it is made up by the chitin. But in case of the animal, such kind of the cell wall, it is totally absent. Next point we are talking about number second property, number second parameter, that is plasma membrane. For the plasma membrane, I'm using just here. Plasma membrane, also called as the cell membrane. So both in case of the prokaryotic system as well as eukaryotic system, plasma membrane is present. Yes, but please remember, whenever we are talking about outermost boundary, that is plasma membrane is present, but membrane bound organelles, they are absent. So plasma membrane present hoga, but membrane bound cell organelles like chloroplast, mitochondria, organelles, they are totally absent. But when you compare this point with eukaryotic system, yes, membrane bound organelles, they are present. Okay, simple one. But we are taking one unique example with respect to ribosome. What kind of the ribosome it is present in case of the prokaryotic system and the eukaryotic system? Because ribosome, these are the also organelle. But please remember, ribosome, these are the without plasma membrane. So next point we are talking about the ribosome, and ribosome, these are the non-membrane organelle in case of the pro as well as in case of the u. So ribosome organelle hai, please remember. But whatever we are talking about the plasma membrane, that plasma membrane part is totally absent. That's why we are talking about ribosome. These are the without plasma membrane organelle. We'll discuss that part later in detail. When we are talking about prokaryotic system, in case of the prokaryotic system, 70S type of ribosome is present. Obviously, because we are talking about the bacteria. So there is a presence of 70S type of ribosome. But when you compare, 8 seconds for loading. Yeah. एक सेकेंड वेदांत, बेटा थोड़ा कहाँ गया? So we are talking about which part? That is part of ribosome. So in case of the bacteria, ribosome is present, that is 70th type of the ribosome, and such type of the ribosome they simply present in cytoplasm. So cytoplasm. Okay? But when you compare this point with eukaryotic system, in case of the eukaryotic system, 70th type of the ribosome also present, as well as 80S type of ribosome also present. Both type of the ribosome they are present in eukaryotic system. 70S type of the ribosome they are associated with the two organelle, powerhouse of the cell, that is mitochondria, and the another organelle, chloroplast. So in whole eukaryotic system, two examples, that is mitochondria and chloroplast, where 70S type of ribosome is present. But when you talk about the 80S type of the ribosome, yes, these are also present. And 80S type of ribosome, they are present in cytoplasm, and as well as they are associated with rough ER, that is rough endoplasmic reticulum, RER. So quickly complete this part. If there is a one question, which type of the ribosome they are present in eukaryotic system, so your answer will be 70S and the 80S. Both answers should be correct. Please remember 70S plus 80S. But अगर पूछा गया है in case of the mitochondria and the chloroplast, which type of the ribosome is present, so your answer will be only 70S. So please read question carefully because statement question होगा definitely. So ribosome, these are the without, it's the organelle but without plasma membrane. In case of the bacteria, normally 70S type of the ribosome is present, and in case of eukaryotic system, both 70 plus 80S type of the organelles are present. Means ribosome is present. Chalo. Got it? Okay. Yeah. So any doubt from today's class?